We analyze DNA through the process of agarose gel electrophoresis. Agarose is a linear carbohydrate that is found from the cell walls of red algae, and it is basically this polymer that is repeating in structure. And what happens is it's soluble in water to some extent. We put it into a buffer like tris borate EDTA or tris acetate EDTA, depending on the buffering capacity we desire. And what happens is, depending on the amount of agar to or agarose to buffer ratio, you have a firmer or looser gel matrix. That means that it creates a webbing when you dissolve the agarose powder into the buffer and melt it with heat, usually in a microwave. When you let it cool down and solidify, you put it into a casting tray, as we see here, and then you put in some combs that will later form these voids for wells. And what you do is you let it cool and solidify, and the carbohydrates basically start to meld together and trap that agarose and form pores throughout this matrix. It's a gel. We load our sample into these wells inside a tank of buffer using something that's heavy and dense that'll help it come down into the wells instead of spreading out. So oftentimes our loading buffer will have glycerol in it to help the samples separate. We can't see DNA. However, if we use some sort of intercalating agent that will bind to the DNA, we can actually visualize the DNA. The agents that we use in labs are usually ethidium bromide, as we see on the left, and unfortunately it is rather carcinogenic. So in the context of educational labs, we don't use it. We tend to use the one on the right, which is called cyber green. It's safe because it does not pass through the cell membranes very easily, as ethidium bromide does. And as we can imagine, if it goes through your cell membranes, it can bind to your DNA and cause a lot of issues. Now, how do we actually identify or study DNA? Well, if you imagine inside the nucleus of a human cell, there are 3.6 billion nucleotides in any nucleus. And when you think about how you can identify various aspects of this, you need a way to manipulate these DNA in some way. And one way that we manipulate them is by using what are called restriction enzymes, or as we see here, are more appropriately called restriction endonucleases. ASE, of course, means it's an enzyme. Nuclease means that it cuts nucleic acids, and endo means within. What does the restriction mean? Restriction means that there is some limitations to how these nucleases cut. There are other standard nucleases, such as deoxyribonuclease, which is just DNase, which will cut DNA anywhere. There are things that are called RNases, which are ribonucleases, and they will cut RNA throughout. And there are very specific ones that we use as tools. It's not useful to just cut DNA randomly, so we restrict them. How do we get these restrictions? Well, that goes back to the origin of where these things come from. First of all, let's understand what it means to digest or cut DNA. The backbone of DNA, as we can see here, is a repetitive sugar phosphate backbone, right? And we always go in a certain polarity of reading DNA and RNA, which is to say 5' to 3'. 5', there should be a phosphate here, and then there's a deoxyribose, followed by a phosphate, followed by deoxyribose, followed by phosphate, followed by a deoxyribose, and so forth. And at the 1 position, we have the nucleobases. In this case, we have a pyrimidine called thymine. In this case, we have the purine called adenine. And we know that there's going to be another chain on this side that is base pairing to these bases here through hydrogen bonding. And we have to remember that that strand is anti-parallel in nature. If we go back to how these things are restricted, we're actually talking about the digestion of the covalent bonds here of the phosphate to the sugar, right? And so this phosphodiester bond will be cleaved, and that's going to be very difficult to do. It requires energy, as opposed to the hydrogen bonds that hold the two strands together are somewhat weak in transit that could be disturbed by just heating it up. So how are they restricted? Well, it turns out that the first restriction enzymes that we were found or discovered were found as defensive mechanisms of bacteria and yeast and fungi and other things like that, where they would actually restrict to specific sequences. And the ones that are very useful for molecular biologists as a tool are those that recognize palindromic sequences. Not all enzymes do this, but the ones that we use as tools, we purposely chose because they're restricted to these areas. So an example of a palindromic sequence, of course, is race car, a man, a plan, canal, Panama, or Civic. And because there are anti-parallel strands, we have to understand that the restriction to these palindromes is the same thing forwards and backwards on one strand versus the other. So this is an example of what that looks like. Here is an example of an enzyme called HindD3. HindD3 comes from a bacterium, and what you can see here is if we follow the 5' strand, so here's 5, and we read it AAGCTT, and then whatever, right, to the 3'. And if we read from this end on the 5', AAGCTT. So it's the same. That's what we mean by palindromic. Remember, the phosphodiester bonds are covalent and therefore difficult to cleave, right? They require energy, and some sort of enzymes will facilitate that. However, these base pairing are held together by weaker hydrogen bonds that can be disrupted just by a little bit of energy, such as heating. HindD3 will break this bond here, right? And you can see it's asymmetrically cutting, right? It's not perfectly in the middle. And what happens is it leaves these overhangs that we have here, right? Such as AGCT, AGCT. And what we know that can happen there is while these things come apart, they are also capable of coming back together very transiently, right? So we call those cohesive or sticky ends. They're sticky because they basically can bind back transiently, and if we use some sort of gluing agent like another enzyme called a ligase, then we can glue them back together, right? Here are two other examples that are common, Echo R1, and as you can see here, the sequence that Echo R1 recognizes is GAATTC, and on this other strand, 5' GAATTC. It does not matter what is the flanking sequence, right? We will always use things that are called 6 cutters or 8 cutters because they're very useful for manipulations of the things that we're looking for, because they will cut frequently but not so frequently as to make many pieces. Once again, you can see that Echo R1 is asymmetric cutter, and therefore it will result in sticky ends. Here, SMA1 cuts CCGGG, or CCGGG. And as you can see, this one is a symmetric cutter. Now, this is different. This leaves what is called a blunt end, right? And a blunt end does not have the ability to have these transient interactions that we have here that lead or guide reassembly if we use the ligase. So therefore, if we had another blunt cutter, such as one called Echo R5, it would actually just as likely bind butt to butt with this piece of DNA here or this piece of DNA here, and if we added a ligase, it would glue them together. And in that case, it would actually destroy those nucleic acid restriction sites, right? Because the sequence will no longer be palindromic for either of those things. So how do we actually analyze those DNA? As we said before, you can set up an agarose gel. And here's the process. You have an agarose gel being poured into a casting chamber. You have combs that leave wells behind. And then we load the samples. We have to make sure that these are in the electrophoresis tank submerged in buffer first before we load them, and then we apply an electric voltage to it and the current will run. Because DNA has phosphate backbones, they are negatively charged, and so therefore we load them towards the negative end because they will be pushed or pulled towards the positive end. And in the first lane, we will have a marker or DNA ladder that acts as a size reference to judge the fragment sizes that we're going to get. Remember, when we poured this, we would have put our intercalating agent in the agarose gel. And as you see here, over time, it has resolved. And here is an image showing it when it's very resolved. We run it as long as we want that we can see the resolution, and we change the percentage of the gel depending on the size of expectation. If it's a very large fragment that we want to separate from other large fragments, we will tend to have a low percentage gel, so things will generally run a little bit faster. If we are looking at very small fragments and trying to differentiate them from each other, we would tend to use a very high concentration gel, such as up to 2%. Anything higher than that would be very unfeasible to run this way, and we would use a different technology, such as polyacrylamide. Um, and here you can see a multi-channel pipette being used to load various samples in a large gel, and we have here the resolution of various bands in our gel. You can watch this video to understand how to do this better, and we'll do this in class.